So, good afternoon, guys. Today we'll be starting with the week 12 of the course host pathogen interaction by Professor Himanshu Kumar. And in this lecture, we'll be mostly discussing about the fungal infection and the protozoan infection. Okay, and the protozoan infection. So, I'm Rishabh, and we'll start the lecture for today. So, starting with the fungal infections. So, as you guys know, that different kinds of fungus are basically available in terms of vinegar, in making vinegar, bread, beer, wine, cheese, and the antibiotics production that is also taken care by the fungal population. So, the products can be toxic, carcinogenic, hallucinogenic, like lysergic acid, LSD, it is used in LSD, coming from rye ergot fungus, Claviceps purpura. Then ringworm, it's a common fungal skin infection that is caused by Microsporum ordoni and Trichophyton tonsorus. Okay, so these are some of the fungal products that can be either toxic or that that we see in our normal life. So approximately 10 to the power 6 people are known to known fungus among that, so 10 to the power 6 are the no nearly 1 lakh fungus species are known, and approx 400 or the 0.04% of the fungus that causes disease to the humans. Okay, then exogenous fungal infection include gain access through injury or inhalation or ingestion. Okay, so how do the fungal infect? They either enter the body through injury or normal inhalation or through food, that is ingestion. Okay, so this is how the normal fungal enters into the body. Then endogenous fungal infection by breaching or gaining access to sterile part of the body. So endogenous fungal infection also takes place, that takes place because of the breaching of the sterile part of the body. Then the overall the fungal diseases are called as mycosis. Okay, so the Latin name for fungi is myco. Okay, so that's why the disease that is caused from these fungus is mycosis. And these fungal diseases or fungal organisms are mostly opportunistic. That means they'll grow when they will grow exponentially whenever their optimum conditions they'll get. Okay, so the infection can be superficial, subcutaneous, or deep in the tissues and system. Okay, so superficial can be above the skin. Same with the cutaneous. Subcutaneous is within the skin. Okay, then in the deep tissues, or it can be systemic. Some infections start from cutaneous and reach to the lungs. For example, coco, dimycosis, that is valley fever. Okay, so they start from the cutaneous route, but they reach to the lungs. Then the fungal infection is associated with acquired or congenital immunodeficiency or malnutrition. Okay, so most of the fungal infections are associated with acquired or congenital immunodeficiency because they'll cause weakening of the immune system, and subsequently they'll try to gain the all the nutrition from the body. That's why they also causes malnutrition. Okay, then talking about the immunity against the fungal infection by the body. So what happens, there is competitive inhibition by commensal organisms, because if individual receive long term antibiotic treatment, they may suffer from oral or vulvovaginal infection by Candida albicans. Okay, so what happens is if we are receiving very long term antibiotic treatment, so in that case what happens, the so in that case the bacteria that we have in the body are actually not there, so they die. Okay, so because of that, there is competitive inhibition, and that causes the sub-infection with Candida albicans. Okay, then neutrophils are important, and neutropenia or malfunction of the neutrophils cause fungal infection. So this shows us that neutrophils are an important immune cells that takes place, or they help in preventing the fungal disease. Okay, then alternative and lectin-dependent complement pathway play important role against fungal infections. So the complement pathways, both alternative as well as the lectin-dependent pathways, play an important role during the fungal infection. So they are first opsonization, and second the membrane attack complex they form. The both of them help in the removal of the fungal pathogens from the body. Okay, so both types of the pathway, the alternative as well as the lectin independent, play into the role. The MBP play important role against C. albicans, C. neoformans, and C. fumigators. So all the three candidas, albicans, neoformans, and fumigators, play has been you are like MBP, that is mannose binding protein, are against all of the three. Okay, then surfactant proteins mainly in the lung play important role again lung fungal infections. So the surfactant proteins that are present in the lungs. So lungs basically the mucus layer. Okay, so they play an important role against the lung fungal infections. Then TLR2 and 4 are also very important. So, as part of the innate immune system, the PRR recognition receptors TLR2 and 4 are help in recognizing the particular fungal PAMs, so that they can actually initiate the immune response. Then T cell plays important role against fungal infections, but not the B cells. So T cell are an important factor that play an important role during a fungal infection, but not the B cell. Okay, so from these things, it comes to notice that neutrophils are important. Both the alternative and the lectin-dependent complement pathways play an important role during the fungal infection. Out of these, the mannose binding protein play an important role, binding to all the species of the Candida. Surfactant proteins in the lungs help in achieving the immune response. TLR2 and 4 are an important PRR of the innate immune system that helps to recognize the fungal infections. And out of the T and B lymphocytes, T lymphocytes an important role in displaying the immune response against the fungal infection, but not the B cells. Okay, so this is how our body actually protects us from the fungal infection. So moving on, the innate immune response, how it takes place for the fungal infection. So talking about this, so first is TLR2. So TLR2 helps in phagocytosis, and it helps in the release of the pro-inflammatory cytokines. Okay, and it recognizes Candida, then it recognizes Cryptococcus, and it also recognizes Paracoccidosis. Okay, then talking about the dectin 1. So dectin 1 is also a receptor that helps in recognizes different fungal pathogens. It helps in activating either the phagocytosis, or it helps in activating the inflammasome. Okay, further helps in production of reactive oxygen species, MHC class 2 loading, and release of the type 1 interferons that help in basically inflammatory response. Okay, then moving on to dectin 2, another family that belongs to the dectin family, and it helps in increase of the TH17 cells. Okay, and along with dectin 1 and TLR2 also help in release of the pro-inflammatory cytokines, so that they can reach to they can increase the inflammation against fungal pathogens. Okay, then we have Minkle, another receptor that increases the pro-inflammatory cytokines, and the Th17, that are the regulatory T cells. Then we have MR. Okay, so this MR also helps in increase in the phagocytosis of the macrophages and increase in the IL-17 production. Then we have CR3 and DC sign. So they both help in phagocytosis and increase in the pro-inflammatory cytokine production. So these are some of the PRRs of the innate immune system that helps in the recognition and the increase in the inflammation. So namely TLR2, Dectin 1, Dectin 2, Minkle, MR, CR3, and DC sign. Okay. Now, here are some of the examples of the different molecules that are recognized by the different receptors. Okay, so here we have IL-1 receptor that recognizes Candida albicans, yeast, and Candida hyphae. Okay, so Candida albicans or uska and its hyphae are recognized by the IL-1 receptor. TLR2 recognizes Candida albicans, Aspergillus fumigatus, its hyphae, and the zymogen. Okay, then Dectin 1, it recognizes the beta glucan of the Candida albicans. Okay, then the CD14, TLR4, MD2, and they recognizes GXM or the protein component of the albican hyphae and the Aspergillus fumigatus hyphae, that is mannan. Okay, then the TLR9 actually recognizes Candida albicans and Aspergillus fumigatus. Okay, now all these are dependent upon the MyD88 dependent signaling, as we have studied before, and it leads to the production of pro-inflammatory cytokines IL-12 by the dendritic cells, induction of the respiratory burst and degranulation, and the T helper cell differentiation. So Th1 cell differentiation is also taken care by this pathway itself. So, out of these receptors, when they recognize a particular fungal pathogen, they lead to the production of pro-inflammatory cytokines, IL-12 production, induction of the respiratory burst and degranulation of the particular base of degranulation, and the Th1 cell differentiation, so that they can lead to the production of they help in the maturation of the TC cells. Okay, so now moving on to the invasive fungal disease by the fungal infection. Okay, so these are some of the dimorphic fungi. That means they uh, they can grow at 25 and 37 both, but the most favorable temperature is 37 degrees Celsius. For example, we have Blastomyces dermatidis, so they can actually grow at 37 degrees Celsius. We have coccidioides imitates that grow as cocci, that means in circles, okay, at 37 degrees Celsius. Then we have Histoplasma capsulatum that also grows in size in 37 degrees Celsius. Then we have Paracoccidioides brasiliensis that also grows at properly at 37 degrees Celsius. Okay. Then we have opportunistic fungi that, for example, we have Candida albicans, that is a budding yeast, pseudohypha or germ tube. Okay, so this can be a hypha. Then we have Cryptococcus neoformans that grows as a capsule. Okay. Then we have Aspergillus fumigatus that have a conidia. So they grow as a conidia and they infect as a conidia. Okay. So these are some of the fungals that are either dimorphic or opportunistic in their growth. Okay. So moving on, how they interact with the dendritic cells, how the fungus interacts with the dendritic cells. So the mouse immature dendritic cells were able to phagocytose different fungal morphocytes. Okay, so these green colored ones are the different fungal morphocytes from different yeast. So this is Candida, then this is hyphae from the Candida, then this was Conidia from the Aspergillus, and this is hyphae from the Aspergillus. And these red color ones are the dendritic cells. So they were able to phagocytose all the hyphae or the conidia of those particular fungus. Okay, through different phagocytic modalities. So they can either uh, using coiling phagocytosis, they can engulf the bacteria or they can engulf the fungus. Then zipper type phagocytosis, coiling phagocytosis, and this one also zipper phagocytosis for the Aspergillus hyphae. Okay, so this leads to increase in the MHC class 2 molecules because they are engulfing the extracellular pathogen. So that's why they will load this pathogen to the MHC class 2 molecules. Okay, then they will increase the co-stimulation based on the B7 CD86 co-stimulation, and it will increase into IL-12 production, IL-4 or 10 production. Then the mostly the conidia leads to IL-12 production, and the hyphae leads to IL-4 IL-10 production. Okay, and they instruct the protective Th1 cell and the non-protective Th2 cells to be responded. Okay, so, as you can see in the conidia case, the Th1 producing hyphen gamma are increased in number in both the cases. Okay, while when the hyphae were treated, the IL-4 producing Th2 cells were high in number as compared to the interferon gamma producing Th1 cells. So, this shows that either conidia from any of these particular fungi give rise to the IL-12 production and IFN gamma production. But the hyphae give rise to IL-4, IL-10 or producing Th2 cells. Okay, so this is the differentiation between the two. Okay, so moving on. Now moving on to how the innate and adaptive immune responses act against the fungal pathogens. Okay, so the innate immunity, as you can see, this is the mucosal surface and this is the fungal pathogen. Now the PRRs get activated and the complement system. So these will opsonize these particular fungal pathogens so that they can be engulfed by the macrophages or the different APCs, antigen presenting cells. The neutrophils come into play. Okay, the innate immune functions such as phagocytosis and neutrophil degranulation takes place. Then they release inflammatory cytokines IL-10, 12, and 18 for the dendritic cell to get maturation, and then they'll mature dendritic cell in the lymph node. They'll present the antigens to the T cell basically through MHC class 1 or 2, and then the degranulation actually the inflammatory response by Th1 increases the IL-12. Okay, so IL-12 basically increases with IFN gamma and TNF from the Th1. IL-4 acts on Th2 for releasing IL-4 and IL-5, and IL-10 will act on the T regulatory cells for increasing TGF beta and the IL-10. Okay, so this dendritic cell actually leads to the increase in the these interleukins from the Ts. Okay, so this is the adaptive immunity that takes place with the help of the innate immunity, the neutrophils and the different TLR containing cells. Okay, So this is the counterplay between the innate and the adaptive immune system when the fungal infection takes place in the body. Okay, so now moving on to the invasive fungal disease by the fungal infection. So previously we have seen that how the different pathogens cause to the invasive fungal disease. So the fungal pathogens are associated with people who are at risk due to underlying health problems. Okay, so whenever there is another health problem that is associated that leads to compromised health condition, okay, that leads to the fungal pathogens to replicate inside the body. Okay, then the weakened immune system, such as chronic lung disease, prior tuberculosis, HIV, cancer, and diabetes mellitus, that weakens the immune system. So this leads to the infection by the co-infection by the fungal pathogens because the immune system is already weakened due to another major infection. Okay, then critically ill patients, as in ICU, patients undergoing invasive medical procedures and receiving broad spectrum antibiotics, and those taking immune suppressing medicines are all at risk. Okay, in these cases. Okay, so these are the major people that encounter fungal infection due to either co-infection or another immune compromised conditions. Okay, and so who or are at risk. So it is rising as the risk population continues to expand due to advancement in modern medicine and the accessibility to therapies. Okay, because most of the therapies actually weakens the immune system rather than boosting it. Okay, then interventions that impair the immune system, such as chemotherapy, immunotherapy for cancer, solid organ transplantation, because in that case we give some immunosuppressive drugs. So in that case, those people are at very high risk. Okay, patients with chronic obstructive pulmonary disease, liver or kidney disease, viral respiratory tract infections such as influenza, and those with prior non-tuberculosis mycobacterium infections. Okay, so these are the major population that is highly affected. The coronavirus disease pandemic has also been associated with the increase in the incidences of comorbid invasive fungal infections. So three groups of COVID-19 associated fungal infection were seen: aspergillosis, mucormycosis, and candidalia. Finally, there is evidence that suggests that both the incidence and geographic range of fungal infection are expanding globally due to climate change. Okay, so the fungus actually needs a humid environment to grow. Okay, so in that case, it actually increases the conditions in all over the world so that they are actually prone to the fungal infections. Okay, now, the fungal infections were enhanced due to rapid emergence of antifungal resistance and, in many settings, limited access to quality diagnostic and the treatment. Okay, so as antimicrobial resistance is actually increasing due to the increased use of the uh, drugs against those pathogens. So this is and the limited access to the quality diagnostic and treatment is a major problem. Then antifungal resistance has major implications for human health. It leads to prolonged therapy and hospital stays because of that. Okay, so this is how different fungal infections are increasing around the world because of the newer technologies and the newer diseases that we are encountering. Okay, so these are the most common groups that have been identified by WHO, that is World Health Organization, and they divided the groups into critical priority groups that includes Cryptococcus neoformans, Candida auris, Aspergillus fumigatus, and Candida albicans. Okay, in them. So these are the groups that have been included in the critical priority group by the WHO. Then we have high priority group that includes Candida glabrata, plant species. We have Eumycetoma causative agents. Then we have mucorales, Fusarium species, Candida tropicalis, and Candida parasilosis. So these are the high priority groups. So after the critical priority groups, that means these must be taken care of immediately in developing the amount of therapies, and these are the high priority groups in that they are growing into number of diseases. Okay, so the next priority was be given to these, and the medium priority group includes uh, Pseudosporium species, Lomentospora prolificans, then Coccidioids species, then we have uh, Pichia cutrivazili, and that is also called as Candida krusei. And then we have Cryptococcus gatti. Then we have Talaromyces marnifei. Then we have Pneumocystis jirvoki, and then we have Paracoccidioids species. Okay, so these are the medium priority groups that we can see in a in the groups that are defined by the fungal pathogens. Okay, now moving on to the next disease, that is cryptococcosis. Coccosis. Okay, so this disease was approximately 1 million of new cases of cryptococcosis occur annually, and mortality rate is 50%. So half of the million cases, half of the million people die because of this. So Cryptococcus neoformans C. gatti occurs worldwide in nature and is isolated readily from a dry pigeon feces as well as trees, soil, and other sites. So the major site from which it can be isolated, these two pathogens, that is Cryptococcus neoformans and Cryptococcus gatti. So these are the dry pigeon feces, trees, soils, and other sites. From the lungs, these neurotropic yeasts typically migrate to the CNS where they cause meningoencephalitis. Okay, so from the lungs in which they have been inhaled, these neurotropic yeast. So why these neurotropic? Because they can easily go into the CNS and they cause meningoencephalitis. Okay, they can also have the capacity to infect many other organs, example skin, eyes, and the prostate. Okay, so they have the capacity to infect many other organs, including skin, eyes, and the prostate. Then we have Candida neoformans occurs in immunocompetent persons, but more often in patients with HIV or AIDS, hematogenous malignancies, and other immunosuppressive conditions. Okay, so these occurs in immunocompetent persons, but more often in people suffering from HIV or AIDS, hematogenous malignancies, and other immunosuppressive conditions in which the Candida neoformans can easily grow without encountering our immune system. Okay, so how does this actually infect into the patients? So avian faces that have Candida neoformans, or the trees that have both neoformans and gatti, they are inhaled or of the desiccated yeast, example basidiospores, to the pulmonary alveoli. So they are inhaled by the human system, and in the alveoli they cause phagocytosis of the alveolar macrophages. Now they lead to resolution, latency, pulmonary disease, and or spread of one or more sites. Now they'll reach to the CNS. Okay. Now there they'll grow. Okay. So this is how they infect and they grow inside the human body. So first they reach the alveoli, they cleave the macrophages, phagocytose them. So the macrophages actually get macrophages phagocytose this thing, and then they from there they actually move to the CNS where they call the encephalitis. Okay, so these are the capsule of the neoformans that can be seen in the pulmonary lavage specimen. Okay, moving on to the second disease, that is aspergillosis. So aspergillosis is caused by a number of aspergillus species, for example, aspergillus fumigatus, the most common form of a human pathogens, and other ones are aspergillus flavus, Niger terrace, and the lenticules. This also can cause the disease. This mold produces abundant small cornelia that are easily aerosoled. So this also is inhaled as an aerosol. So in immunocompromised patients, especially those with leukemia, stem cell transplant patients, and individuals taking corticosteroids, the conidia may germinate to produce hyphae that invade the lungs and other tissues. So these are majorly infecting the patients that are immunocompromised or those have leukemia, stem cell transplant, and individuals taking corticosteroids. The conidia they germinate to produce hyphae that invade the lungs and other tissues. So the hyphae majorly invade the other tissues. Okay. In the lungs, the alveolar macrophages are able to engulf and destroy the conidia. However, macrophages from corticosteroid treated animals or immunocompromised patients have a diminished ability to continue the inoculum. Okay, so in the lung, conidia swell and germinate to produce hyphae that have a tendency to invade pre existing cavities, that is, aspergilloma or fungus balls or blood vessels. Okay. So the macrophages, the alveolar macrophages, have the ability to engulf and destroy this pathogen. But the macrophages from corticosteroid treated animals or immunocompromised patients have that ability diminished. So in that case, what happens? The conidia swells up and germinate to produce hyphae. Now, this hyphae can actually invade the pre-existing cavities such as aspergilloma or fungus balls or the blood vessels. From there, they can easily infect the other tissues. Okay, so these are the different images from the stain of the aspergillus species. So here they are they stained with gomori methane silver, and these are also the hyphae of the different aspergillus species. Okay, now moving on to the parasite infection. So the parasitic infection are caused by either the protozoans or the helminths or the worms. Okay, so the protozoans are unicellular eukaryotes. They can replicate within the host and they move based on means of the locomotion. Okay, so the type of parasites are based on the mode of locomotion they have. So they can be flagellates, example trypanosoma or leishmania, or they can be amoebae. Okay, amoebic. That means they have a false feet, and they have entamoeba histolica. Then they have the sporozoa, for example, Toxoplasma gondii and the Plasmodium species. So, and the last one, they have ciliates that have cilia as their movement. So they have Bellentidium coli. Okay, so these are the different types of protozoa that is based upon the locomotive pattern that they have. Then we have the helminths or the worms. These are multicellular organisms. They infect to produce their to reproduce on. They infect humans, but they reproduce their own. And there are two phyla, nematoda that is roundworm and the plethelminthes that are the flatworms. Okay, so the nematodes that infect the human body is Ascaris lumbricoids, and the plethelminthes that we have is Psych, Cyclostoma monsoni and the Tinea solium. Okay, so these are the flatworms. So these are the example of the protozoans and the helminths that majorly affect the human body. So we have the case fatality rate that is common for the parasitic uh, infections. So the case fatality rate is defined as the number of deaths upon total number of the cases that have been reported into 100. So this is recorded as a percentage. Okay, so in case of the case fatality rate, the uh, Organisms that is below 10 is the first number is uh, schistosomiasis, guinea worm disease, malaria, then echinococcus, that is cystic, and the leishmaniasis. Okay, so these five diseases are basically have a CFR that is very low. That means the number of deaths are cured. Okay, so that are very less as compared to the number of cases that are reported. Then we have the moderate or high in case of leishmaniasis, that is visceral. Then we have sleeping sickness if untreated, and the uh, echinococcus, iconococcis, that is alveolar. Okay, so in this case, this is the CFR is really high. Okay, then it is an epidemiological metric that measures the proportion of people who die from a specified disease among all positive cases over a period of time. This metric actually indicates the severity of the disease and is often used as a prognosis. Okay, so this is how we check for the fatality of the disease, and then if we can use it as a prognosis. Okay, so then we have the average basic production number of a common parasitic infection. So it is also a epidemiologic metric that describes the transmissibility of the infection agents. Okay, so how it gets transmitted. So R naught basically measures the expected number of secondary infections produced by a single infectious individual in a susceptible population during a mean infection period. So it actually checks how a single infectious individual can actually spread the particular disease in a population during a mean infection period. So this is how the average basic number reproduction number is defined. So in that case, the leishmaniasis, echinococcus, sleeping sickness, helminthiasis, and schistosomiasis are they have a very low R naught. But in case of guinea worm disease, lymphatic filariasis, and the malaria, the R naught is typically high, that is more than 10. Okay, and these star marked diseases are basically caused by the worms, or sorry, or transmitted by the mosquito bites. Okay, so this shows that some of the diseases like leishmaniasis and sleeping sickness, they have a very low infectious rate or the mode of transmission as compared to malaria, or as compared to malaria or lymphatic filariasis that have a high transmission rate from the infected individual. Okay, moving on to the main disease, that is malaria. Okay, so malaria is caused by protozoan pathogens of the Plasmodium species. So Plasmodium, talking about Plasmodium, is a single-celled eukaryotic organism that belongs to the phylum of Epicomplexa, which is named after the apical complex that is involved in the host cell invasion. Now, the initial symptoms include fever, headache, shivering, and vomiting if not treated early. And depending upon the species responsible for the disease, severe anemia, metabolic acidosis, heart failure, burden on kidney, and the cerebral malaria may occur, and then it may lead to death. So these are the common symptoms of the malaria. So the initial ones include severe fever, headache, shivering, vomiting, and if they are not treated, then they can be severe anemia, metabolic acidosis, heart failure, burden on kidney, and the cerebral malaria following the death. So, for the discovery of the malarial parasite in the gastrointestinal tract of the Anopheles mosquitoes led to the realization that malaria was transmitted by the Anopheles mosquito, and this was studied by Sir Ronald Ross, and he received the Nobel Prize in 1902 in physiology. Then, secondly, Sir Leverin showed that the malaria is caused by a single-celled organism, that is a protozoan of the Plasmodium family, which attacks RBCs. Leverin also identified that a single-cell parasite that cause other diseases also, and he received Nobel Prize in 1907. Okay, because at that time malarial research was top notch. So that's why all the people focused on how these organisms actually grow and how they infect other people as well. Now talking about the main causative agent of malaria, that is the plasmodium species. So five human infective plasmodium species are listed here. First is the falciparum, the most common and the most causative one. So it causes the bulk of malaria associated morbidity and mortality in sub-Saharan Africa. Okay, so the major region where the malaria is spread is the sub-Saharan Africa. So Plasmodium falciparum is associated with severe malaria and complication in pregnancy. Most malaria-related deaths are associated with these species. Then we have Plasmodium vivax. So it accounts for majority of the malaria cases in Central and South America and in temperate climates, that is Southeast Asia and the Ethiopia. So vivax actually cause malaria cases in Central and South America and in some of the temperate climates like Southeast Asia and Ethiopia. Then we have the Plasmodium ovale. So ovale is found in Africa and Asia and it's prevalent in West Africa. Then we have the Plasmodium malariae that cause the mildest infections, although it has been associated with splenomegaly and or the renal damage upon chronic infection. Then we have Plasmodium knowlesi. So initially considered as a parasite of non-human primates, cannot only cause malaria in humans but can also lead to severe and even fatal malarial complications. Okay, so these are the five Plasmodium species that have been identified till now. Okay, so now and how they cause different malaria in different regions with different intensities. Okay, so now moving on to the life cycle of malaria. So so here is the life cycle of the malarial pathogen in the human cells. Okay, so starting with the infective stage, that is the mosquito takes a blood meal and injects the sporozoites into the human. So it takes the blood but injects the sporozoite into the human. Now these sporozoites travel through blood to the liver cell and they start uh, exponentially increasing inside the infected liver cell. Now they grow and become a schizont. Okay, so now this schizont becomes growth and it leads to the ruptured schizont and this release the merozoites. Okay, so this is a exoerythrocytic cycle in which it goes into the infected liver cell followed by its production as a merozoites. Okay, now we have the blood stages in the humans. So this merozoites now infect the RBCs. Okay, so this is called as erythrocytic cycle. So now it infects the RBCs, and in the RBCs, it includes the immature trophozoite, that is a ring stage. Okay, so after the mature trophozoite, so it actually goes into gametocytes, that is a male and a female. Okay, so after that they grows into the male and female and the schizosome is ruptured into it moves into the ruptured schizosome and then it can actually move to the different stage where it will be infected by the merozoite, so it actually is increasing in its number. Okay, so now at this stage, it is the diagnostic stage. The diagnostic that takes place for the malarial pathogen inside the human is the schizosont in the erythrocytic cycle. Okay, now these gametocytes actually turn into the falciparum, that is the male and the female gametocytes. Okay, and these are the microgametes here. So these microgametes now are taken by the mosquito taking during the blood meal and it injects the gametocyte into the mosquito. Okay, so now the gametocyte actually move to the sporozoites inside the mosquito. So that is a breeding cycle inside the mosquito. So from sporozoites, it moved to the merozoites, and the merozoites moved to the RBCs. From there, they increase in number inside the gamete, in form of the gametocytes. Now these gametocytes are taken by the mosquito again, and these gametocytes fuse and produce sporozoites inside the particular mosquito. Okay, so now these gametocytes fuse and form a zygote. Okay, so we have a macrogametocyte and a X-flagellated microgametocyte. So this zygote leads to ookinete that invade the midgut wall, that leads to the oocyte and the mosquito stages, the ruptured oocyte that release the sporozoites. Okay, so this is how it takes place in the, this is called as the sporogenic cycle that takes place in the mosquito. So this is how the plasmodium species actually infect the humans, and it replicates in humans followed by the mosquito. Okay, so now moving on to the life cycle in a mosquito. So it basically happens in the gut of the mosquito. So the in mosquito's gut, the male gametocyte produces 8 microgametes after three rounds of mitosis. So one male gametocyte produces 8 gametes by 3 rounds of mitosis, and the female gametocyte matures into the macrogamete. Okay, so that is a macrogamete. Now the male gametocytes are motile; they form with the flagellate and seek the female macrogamete. The male and the female gametocytes fuse, forming a diploid zygote which elongates into an ookinete. Okay, so now the male and female gamete fuses, forming a zygote that elongates into an ookinete. So this is a ookinete. Okay, now this motile form exits from the lumen of the gut across the epithelium as an oocyte. Now oocyst. So this will now release into an oocyst. Now the oocyst undergoes cycle of replication and forms the sporozoites. Okay, now these elongated forms are called as the sporozoites, which move from the abdomen to the salivary glands. So these are the salivary glands of the mosquito, and these oocysts move to the sporozoites, and these move from the abdomen to the salivary glands. Okay, now after 7 to 10 days, the mosquito feeds on blood containing gametocyte. It may be armed and able to infect another human with the plasmodium species with her bite. Okay, so after 10 to 14 days of taking the blood meal, it can easily inject the full modified sporozoites into the humans. Okay, so that is actually loaded plasmodium species that can cause malaria into the humans. Okay, so this is how it takes place into the humans. So now the mosquito has taken the blood meal from human that have the male microgamete and the female macrogamete. The female macrogamete has a macro oocyte. Okay, and then the male gamete fuses with the macrogamete, forming a zygote that elongates into the ookinete. The ookinete becomes motile and forms a oocyst. This oocyst develop into sporozoites that travel from the abdomen to the salivary gland. Now, this salivary gland will now give a blood meal. During the blood meal from humans, it will inject the sporozoites into the humans. So, this is how the life cycle takes place in a mosquito. So, the malarial symptoms include recurrent chills, fever, and sweating, followed by symptoms that peaks every 48 hours. Then we have weakness, anemia, splenomegaly due to blockage of the blood capillary by the merozoites. Then we have damage several vital organs, for example, cerebral damage, heart failure, burden on kidneys, and the renal failure, because they are transmitted to different parts of the body through blood. The symptoms of malaria is not due to Plasmodium species, but it due to overproduction of TNF alpha because cancer patients treated with TNF alpha show malaria like symptoms. Okay, so the major symptoms are not caused by the plasmodium but because of the immune response that is produced because of the infection of plasmodium species inside the body. The host response to plasmodium include innate immunity that provides the initial defense against the infection when pathogens are first encountered, which is followed by the development of specific antibody secreting B cells. Then, children aged under 14 years have lowest immunity to malaria as they produce low amount of antibodies against the merozoite form of the plasmodium. It is believed that the pathology associated with the malaria is consequence of an imbalanced cytokine secretion in which the pro-inflammatory cytokine responses dominate. So that leads to the symptoms of the malaria. Then TLR9, a nucleotide sensing receptor, is involved in the host immune response against plasmodium. Then TLR9 induced high levels of TNF production leads to clinical symptoms such as fever. Okay. In addition, the membrane of the infected RBCs stiffens, and this loss of deformity contributes to the obstruction of capillaries, which has life threatening consequences in severe malaria cases where the vital organs are affected. Okay, so the take home message is the symptoms of the malaria are not caused specifically by the plasmodium species, but how the body responds to it. Then the TLR9 antibodies and the TNF alpha are the major causes of that provide the immune system and they stop the fever. They are cause of the symptoms of the plasmodium. Okay. Now there are certain drugs that are that are for the plasmodium. So we have so these are the different compounds that are against the plasmodium species. So first we have the aryl amino alcohol compounds that includes the quinine family. Then we have the antifoliate compounds like methamine, proguanil, chloroproguanil, and trimethoprim. Then we have artemisinin compounds that includes artemisinin, dihydroartemisinin, and artemether. So chloroquine works by interfering with heme dimerization, the detoxifying biochemical process within the malarial parasite that typically yields the malarial pigment hemozoin. The artemisinin was first isolated from the stems, leaves, and flowers of Artemisia annua by the Chinese scientists. And this artemisinin effectively kills the malarial parasites within host erythrocytes, after which the dead parasites are culled by the spleen, formally infected RBCs intact and thus they are circulating inside the body. Okay, so this is how different drugs acts on the Plasmodium species. Now we move on to the second disease that is the African sleeping sickness. Okay, so African trypanosomaniasis African trypanosomoniasis, also known as sleeping sickness, caused by a microscopic parasite that is Trypanosoma brucei. So these are flagellated protozoans, causes chronic and debilitating disease characterized by a unique mitochondrial DNA and lives exclusively as an extracellular parasite. There are two types, saliva type that is salivarian and the fecal contamination that is caused by stercorenia. Okay, so it is divided into two stages. First is early stage, the parasites can be found in the bloodstreams and interstitial spaces of several organs. Patients experience myriad symptoms, including chronic and intermittent fever, headache, pruritus, lymphadenopathy, and hepatosplenomegaly in the early stages. Followed by the parasite actively invades the CNS, making the start of the late stage. Sleep disturbances, neuropsychiatric disorders dominate the clinical presentation, giving rise to the disease common name of the sleeping sickness. So they'll not be able to sleep properly in the later stages. Then Trypanosoma brucei gambesi infection is chronic with an estimated average duration of about 3 years, evenly divided between the two stages. And then another species, that is Rodensians disease, is usually acute and death occurs within weeks to months, possibly due to a parasitic infection being less adapted to the humans. Okay, and it is transmitted by the Discus fly, that is Glossina species, which is found only in the sub-Saharan Africa. So there are two morphological distinction species that can cause distinct disease patterns in humans. First, we have the Gambesi that cause slowly progressing African trypanosomiasis in Western and Central Africa, and we have the Rodentians that causes a more acute African trypanosomiasis in Eastern and Southern Africa. So that's why this is more deadly as compared to the Gambisi one. Okay, so as Plasmodium, it also follows two major infection cycles. First is in the humans. So when the fly takes a blood meal and infects the metacyclic trypanomastigotes into the humans, so now the infected metacyclic trypanomastigotes transform into bloodstream trypanomastigotes, which are carried to other sites. Okay, so now they multiply by binary fission in various body fluids, example blood, lymph, and spinal fluid. Then the circulating trypanomastigotes in blood during acute phase, usually undetectable in the latent phase. Okay. Then the testis fly takes this bloodstream trypanomastigotes, which are ingested. Okay. So after this, these trypanomastigotes transform into procyclic tryptomastigotes in the vector midgut. These procyclic trypanomastigotes multiply by binary fission, so they divide into the mosquito, and then procyclic trypanomastigotes leave the midgut and transform into the epimastigotes. These epimastigotes multiply in salivary gland. They transform into metacyclic trypanomastigotes. Now, these metacyclic trypanomastigotes are the major infection parts. So now these are then infected again into the humans, where they increase in number. Okay, then the cattle and possibly wild ungulates are reservoirs for the uh, Trypanosoma rhodesian. Okay, so these are the reservoir for this Trypanosoma rhodesians. Okay, now moving on to the cycle in the mammalian host and the testis fly. So they have the cell proliferation in this slender. Then they have the parasitomania, in which the environmental sensing and density dependent differentiation takes place, followed by the morphological and gene expression changes. Then they have the survival in the host. Okay, so this is called as cell quiescence. So now the cell quiescence, we have the survival of the vector, and the morphological and gene changes lead to procyclic, that is cell proliferation. Okay, then after this they lead to the immune clearance and the senescence, and they lead to the cell proliferation or the slender form of the particular trypanosome. Okay, So, how this trypanosome resistance to the mammalian immunity. When the parasite is phagocytosed, stress-induced activation of the adenylate cyclase of the trypanosome plasma membrane leads to the parasite releasing cyclic AMP into the myeloid cells, and the consecutive activation of the protein kinase A, inhibition of the TNF synthesis, thus preventing the immediate elimination of the trypanosomes by the mammalian host. In the mammalian host, the whole parasite is covered with a coat of about 10 to the power 7 identical molecules of glycoprotein, the VSJ, okay, which is anchored into the cell membrane via a glycolipid, glycosyl phosphatidyl inositol, that is GPA. The parasites are able to frequently switch their glycoprotein coat, continuously making the antibody response of the host obsolete and evading the clearance. So this is how it able to resist the mammalian immunity. Okay. Then the drugs that are associated with Trypanosoma brucei are they are first removed from the blood and lymph by the use of suramin or pentamidine, and subsequently from the extravascular spaces of the CNS and the CSF with the help of melarsoprol. Other drugs that are effective are eflornithine and nifurtimox. So these are the true drugs that are also effective against the trypanosoma. So the main drugs are suramin and or pentamidine, and the other one is melarsoprol. So these are the three drugs that are usually used to treat trypanosoma based in infections, especially in sleeping sickness. And then we have the eflornithine and the nifurtimox that is used in special cases. Okay, so moving on to some of the questions that can be asked in such cases. So, in the context of malarial transmission, which of the following statements accurately describe the role of the sporozoite? So, what can be the correct answer? So let's see the options. So the question states that what is the correct role of the sporozoite? So it is the infective form found in the bloodstream of the host. It is formed during the sexual reproduction phase in the mosquito. No. It is formed that invades the liver cell after being inoculated by the mosquito. It causes the clinical manifestation of the malaria. It undergoes asexual multiplication in erythrocytes. So the correct answer is option 3. That means it invades the liver cells after being inoculated by the mosquito, as we have seen in this case. So the spirochetes are the main molecules that are infected by the mosquito into the human body. They then infect the liver cell, where they are multiplied, grown in number, and then it moves to the cytosol. Okay, so this cytosol, after growing into merozoites, will now affect the RBCs. Okay, so the correct answer would be option number 3. Now moving on to the next question, which complement pathway is particularly important against fungal infection? So what can be the correct answer? So, as we have studied before, both the alternative and the lectin pathways are involved in against the fungal infection. Okay, so answer is 4. Moving on to the next question. Which phase of the malarial parasite's life cycle is primarily responsible for causing clinical symptoms in the humans? So, what can be the correct answer? So, in the liver stage, it basically transforms from sporozoid to the merozoid that now moves to the RBCs. Okay, in the gametocyte state, it is infected into the mosquito for multiplication. It infects the liver. In the erythrocyte cycle, when it increases in number into the gametocytes, then it causes the rupture of the RBCs. Now, here it causes the uh, clinical symptoms to appear. Okay, so the correct answer is option number 4. Then the ookinete stage takes place in the mosquito. So it will not be appearing in the clinical symptoms. Okay, now moving on to the next question. Which of the following factors contribute to the evasion of the host immune system by the Trypanosoma brucei? So, what can be the correct answer? So the evasion of the immune system by the Trypanosoma brucei is taken care by the variant surface glycoprotein, that is the VSG switching. Then the stress-induced release of the cyclic AMP, the antigenic variation, and the inhibition of the cytokine synthesis. Okay, so these are the different forms how they evade the immune system. Okay, moving on to the next question. Which of the following is the cause of the ringworm infection? So, what can be the correct answer? So, it is caused by a parasite infection. Okay, so the correct, sorry, a fungal infection. Ringworm is a fungal infection, not a parasite infection. So, it is caused by fungus. Okay, so the correct answer is option number three. Let's move on to the next question. How does prolonged immune suppression affect the host's response? So, what can be the correct answer? So, the immune suppression can enhance the immune regulation, reducing fungal virulence? No. It weakens antiviral defenses while sparing antibacterial immunity. Then it disrupts multiple immune pathways, increasing susceptibility. So it can be correct. It mainly compromises bacterial community with minimal fungal impact. No. Then it induces protective compensatory immune responses. No. So the correct answer is option number three. So what happens because of the immune suppression? The immune responses are basically affected. So they increase the susceptibility to various infections. Okay, now let's move on to the next question. What does the term fungal morphogenesis refer to in the context of virulence? So, what can be the correct answer? So, the ability of the fungi photosynthesize? No. The changes in the physical structure of fungi that enhances its pathogenicity? Yes, it can be correct. The genetic makeup of the fungal species? No. The classification of fungi based on their reproductive methods? No. And the interaction between the fungi and other microorganisms? No. So the correct answer is option number 2. So the fungal morphogenesis refers to the changes in the physical structure of the fungi that could enhance their pathogenicity. Okay. Moving on to the next question. Salivarian and stercocarrian modes of transmission are of which of the following? So what can be the correct answer? So they are the forms of the trypanosoma. Okay, so the correct answer is two. So they are the different modes, that one in saliva. Okay, so the salivarian mode in the saliva, and the other one was through feces. Okay, so this is for the trypanosome. So let's move on to the next question. Which Plasmodium species is most commonly associated with severe malaria and high mortality rates? So what can be the correct answer? So the correct answer is option 4, that is Plasmodium falciparum, that causes severe malaria and the high mortality rates. Okay, out of the following. Now let's move on to the next question. Which statements are true regarding the life cycle and infection process of Trypanosoma brucei? So what can be the correct answer? Okay, so let's see the options. The parasite can only survive in human host. No, it can survive in the testis fly also. Then the metacyclic trypanosomacode is injected into humans through the testis fly bite. Yes, this is correct. The infection progresses to two distinct stages with the systemic and the neurological symptoms. Yes, this is also correct. The Trypanosoma brucei has a simple life cycle involving only one host. No, this is false. Okay, because it has two hosts, that is human as well as testis fly. The parasite's genetic adaptability is largely due to its unique mitochondrial DNA. Yes, this is correct. It is primarily transmitted through contaminated water resources. No, this is false. The understanding the life cycle is crucial for developing effective control measure. Yes, this is correct. The presence of surface glycoprotein variation complicates immune detection. Yes, this is also correct. So the correct options are 2, 3, 5, 7, and 8. So this must be 5. Okay. So let's move on to the next question. Which of the following statements are correct? So what can be the correct answer? So let's see the options. Toxoplasma gondii is a fungus that infects most species of the warm-blooded animals. Okay. Ringworm infection is caused by a fungus. Yes. Mycosporium ordini causes valley fever. Then fungal ligand chitin can directly bind to the TLR2. Fungal infections are predominant in HIV infected patients. Yes, this is correct. This is also correct. Please B cell play a major role against fungal infections, not the T cells. No, T cells may play a major role in the fungal infection. Neutrophils play an important role in the fungal infection. Yes. IL-12 is produced in the dendritic cells upon fungal infection. So yes, and then IL-10 is produced in the dendritic cells upon fungal infection. So this is also correct. So the correct options are 2, 4, 5, then 7, 8, and 9. Okay, so these are related to the fungal infections that takes place in the body. Okay, so let's move on to the next question. Which statements are true regarding the parasitic infections? So what can be the correct answer? Okay, so let's see the options. Trypanosomes can frequently switch their protein glycoprotein code. VSJ in the trypanosome is anchored to the cell membrane via a glycolipid and glycosyl phosphatidyl inositol. Yes, this is correct. The TB Gambesi infection is usually acute and death occurs within weeks to months. No, this is for the uh, Rumbesi. Plasmodium are single-celled prokaryotic organisms. No, they are eukaryotic organisms. The symptoms of malaria are due to Plasmodium species, but it may be due to the overproduction of the TNF alpha. Yes, this is correct. Plasmodium vivax, the bulk of causes the bulk of malaria-associated morbidity and mortality in sub-Saharan Africa. So yes, this is correct. Chloroquine effectively kills malarial parasites within the host erythrocytes. Yes. Artemisinin was first isolated from the stems and leaves of the flowers of Artemisia annus. Yes, this is also correct. In the mosquito's gut, the male gametocyte produces 12 microgametes after 3 rounds of mitosis. No, it was 8 gametes. Okay, so the correct answer is 1, yes, 2, then 5 and 9. No, it would be 7 and 8. Okay. Hmm. So it produces 8 microgametes. Okay. So okay, so this was the last question for today. If you guys have any doubt, you can ask. If no doubts, then we'll close the session for today. Thank you guys for joining in.